Okay, so what we've covered so far in Chapter 4 has to do with cell structure and characteristics, cell size, prokaryotic cells, eukaryotic cells, organelles, extracellular matrix, stuff like that. The latter half of Chapter 4 focuses on still cellular characteristics, but sections 4.10 to 414 focus on membranes, cell membranes, and in particular membrane transport, how substances move back and forth across cell membranes. So we're going to tackle that. And again, this is extremely important for you, especially if you're moving on into the health sciences, nursing or dental hygiene or whatever the case may be, because so much of our physiology, so much of our cellular chemistry is taking place at our cell membranes. So this is stuff that you really need to know. Okay, earlier in your textbook, earlier in the chapter, it talked about membranes, membrane structure, and I kind of skirted over that a little bit, and I'm focusing on that now. So in particular, let me take a look at this. Talked about membrane, the plasma membrane in section 4.2. So some of what I'm going over here for a few slides has to do with section 4.2. But then we're going to jump into specifically section 4.10 where it talks about diffusion and work on from there. But as kind of an introductory slide for this whole membrane transport topic, let's take a look at this computer image that I've got on your screen. And it's a computer model of what a plasma membrane looks like. And so because the plasma membrane is the boundary that separates a living cell from its environment, kind of like our atmosphere around our Earth is what separates our living Earth. Of course, the Earth isn't really alive, but all the life that exists on this thin skin on the surface of our planet from outer space. The atmosphere is what is the boundary that separates us from that void of space. So a plasma membrane is a boundary that separates a living cell from its external environment, its surroundings. And plasma membranes, any membrane for that matter, cell membrane, exhibits selective permeability. So you need to know what that term means. You need to be able to use that term. If a membrane is selectively permeable, it means that some substances can cross the membrane, can go back and forth across it, but others can't. So there has to be some property or characteristic, of course, that's causing this. And, we'll go over that. But what other barriers can you think of that are this way, that allow some things to cross but not others? I mean, certainly international borders are like that. You need to have a passport to get across, or be a citizen of a country to get across. Maybe if you're going into a nightclub that is, 21 years and older, then you've got a bouncer there at the front door, and that is, what determines the selective permeability into that nightclub is your age. Or maybe a doorway, you could push chairs through it in your home, but you wouldn't be able to push a piano through your doorway. So there, the selective permeability of your doorway would be based on size, not age, but size. So what kind of characteristics of our plasma membranes are determining this selective permeability? Well, let's come back and look at this computer-generated image. What do the yellow spheres represent? Those would be the hydrophilic heads of the phospholipids. The hydrophilic heads of the phospholipids that contain the phosphate group and the nitrogen choline group. What about the green areas? in between those yellow spheres. Those are the hydrophobic tails, the fatty acids in between. It's kind of like a sandwich. And the blue ribbons in this model are aquaporin proteins. And they allow what kind of molecules through? Aquaporins. They allow water molecules through. And so plasma membranes are not very permeable to water unless there's a special protein put in the membrane called an aquaporin that allows the water molecules to pass through channels, which that leads us to what are the little red and grayish things that are outside the membrane on each side. Those are the water molecules. So if you now look really carefully at this figure, you can see that this computer-generated model is showing these water molecules moving through the aquaporins, moving through those blue ribbons to get across a membrane. So our membranes are selectively permeable, and we're going to go over that in more detail. But again, what's being shown on this computer image is that, yeah, water molecules can't get through the phospholipids, so there has to be a special protein put in there to allow water to go back and forth. So let's dive into this, you guys. We'll go on, move on to the next slide. All right, so let's take a more detailed look at membrane structure, how membranes are put together. So phospholipids. Phospholipids are integral to this. We covered phospholipids back in chapter 3, and here they're rising up again. They're super important. They're amphipathic. If something is amphipathic, it means that it has like opposite characteristics, all in the same entity. So phospholipids are amphipathic because part of the phospholipid is hydrophilic, and part of the phospholipid is hydrophobic. So that makes them amphipathic. The hydrophilic parts are the hydrophilic heads with the phosphate groups that are charged, and the hydrophobic parts are the fatty acids that are nonpolar. So take a look at the figure on the right-hand side of the slide. We're using that little phospholipid symbol. And you can see that they're arranged in a bilayer. There's 2 layers of phospholipids. So membranes, cell membranes are made of a phospholipid bilayer, 2 layers. And the phospholipids spontaneously, means they do it all by themselves because of physical and chemical properties, spontaneously arrange themselves into this bilayer because the hydrophilic heads are pointing towards water, both outside the cell and inside the cell. And so it takes 2 layers of phospholipids in order to have hydrophilic heads pointing towards, opposite sides where there's water on one side and water on the other side. So the phospholipids automatically, spontaneously, all by themselves, form this bilayer. Okay. Now the consistency of phospholipids, it's kind of useful, I think, to have this concept in your head, is a lot like olive oil. So if you had a little beaker or test tube of phospholipids and you were swirling it around, it would look kind of like olive oil, kind of viscous, not super thick, So our membranes then, our cell membranes, are really very oily in a way. They're kind of liquidy, okay, with these phospholipids moving around, billions and billions, trillions of phospholipids in a membrane. So they're like an ocean of phospholipids. That's the fluid component of our membranes. But then scattered amongst those phospholipids are proteins. Proteins are embedded, are stuck into the membrane. And we describe, when I say we, I mean biologists describe the structure of cell membranes as being a fluid mosaic. It's called the fluid mosaic model of membrane structure. And what that phrase refers to, you need to understand this, is that our membranes are made of a fluid of phospholipids with a mosaic pattern of proteins scattered around. So if you look at the bottom part of the figure on your slide, it is representing this fluid mosaic, the oceans of phospholipids that are like olive oil with scattered proteins embedded in that membrane. And the hydrophobic parts of those proteins are going to be kind of hidden from the watery environment outside the cell and inside the cell. So those parts of the proteins that are embedded in the membrane are going to be hydrophobic. That makes them match up to the hydrophobic fatty acids. Whereas the parts of the proteins that are coming into contact with water on the inside and the outside of the cell, those parts of the proteins are hydrophilic. Everything is being constructed and designed in a way because of the physics and the chemistry of our cellular environments. Okay, once again, even though I've got this labeled section 4.10, and it does talk about membrane proteins, but I might talk about it in a little more detail back in that section 4.2. So if you wanted to go back and read, and I had you fill out reading questions, right? So you have some notes from reading from back then. Go look at them. You should review those notes. They should go along kind of with some of this stuff. So here we're kind of taking a step back and going, hey, let's take a look at this world, this universe where two cells are right next to each other, side by side, two animal cells. And what's going on in the membranes of these two cells, especially? What's going on in the membranes? So, of course, the outer membrane, the outer boundary of each cell is the plasma membrane. So you can see that in this figure. There's a plasma membrane for each cell. Each one is made of the phospholipid bilayer, which is selectively permeable, allows some things through and some things can't get through. And each has a mosaic, each phospholipid, each plasma membrane has a mosaic of proteins scattered around. Now, what I'm really getting at here is that each of these proteins, of course, has a specific function, has a specific job it's doing. And the bottom part of the slide is summarizing 6 functional types of proteins that our plasma membranes have in them. And your textbook gives explanations for these, but this is sufficient for you to know here. So you need to know these 6 functional categories and be able to briefly describe, like I do here, what each category of protein does. So compare what I have there for notes with the figure, because they match up. You've got attachment proteins. So those are proteins that specifically are linking the cytoskeleton to the extracellular matrix. So they're sort of a conduit. So you can see them there in the figure, attachment proteins. Receptor proteins, those are receiving signals from outside the cell. Receptor, receiving signals, like hormone signals. Transport proteins, those are proteins that are helping to move substances, solutes, chemicals in and out of cells, transport. Junction proteins, those are proteins that are helping cells to attach to each other, like tight junctions, gap junctions, desmosomes. Those were reviewed in the last narration in this chapter. Glycoproteins, those have sugars that are sticking out and they're used as identification tags for other cells to recognize them. And lastly, enzymes. These are proteins that are helping to speed up or accelerate chemical reactions. So there you go. There's the quick overview. Be familiar with these 6 categories of proteins that are found in our membranes and what they do. And really take a close look at this figure and have it sink in. Even do a sketch, do a drawing of this figure on a blank piece of paper to help you learn. I mean, I've got, of course, a worksheet that you're working on here. So yeah, you're taking notes. So let's touch on the topic of membrane fluidity, how fluid membranes are. Remember I said a couple slides ago that phospholipids are like the consistency of olive oil? That's true, but based on how the phospholipids are put together, like what kinds of fatty acids make them up, that contributes to how fluid the membrane is. Another component is cholesterol. Cholesterol is found scattered amongst the phospholipids, and that also controls how fluid the membrane is. So let's look at some specifics here, okay? Heat energy makes the phospholipids move around, shifting positions with one another like people in a crowd. So their heat energy makes the phospholipids vibrate and move, and they're shifting around literally like millions and millions of people in a crowd. So they're shifting laterally, like with other phospholipids, 10 to the 7th times per second. It's insane how quickly these phospholipids are moving around. But they only flip to the other side of the membrane, any particular phospholipid, like once a month. Wow. So why is that? The top figure is kind of showing you this. Well, because that heat energy very easily makes the phospholipids just shift around with each other in the same layer, like people in a crowd. But for a phospholipid to flip and go to the other side of the membrane, that would require that hydrophilic head that's pointing out towards the water to move away from the water and migrate all the way through the membrane and then point to the other side towards the water, if you can envision that. And that's just very unlikely to happen. So the phospholipids are very stable in their own side of the membrane, in their own layer of the bilayer, moving around a lot, but they're not flipping back and forth very often. Okay. Now, what can control the fluidity of the membrane? Well, one major thing is whether the fatty acids are unsaturated or saturated. And that's the middle part of the figure I have on this slide. If phospholipids are made mostly or have a lot of unsaturated fatty acids, that's going to make them more fluid. They're not going to be as packed together. They're going to move around more. If the phospholipids are made with saturated hydrocarbon tails, fatty acids, they're going to pack really close together and it's going to be more solid, not as fluid. So that's a major point. And then finally on the bottom there, cholesterol acts as a fluidity buffer. So at high temperatures, cholesterol helps to decrease phospholipid movement, keeping the membrane more viscous. And at low temperatures, the cholesterol impedes or blocks the phospholipids from packing close together, like nudging close together. And so that keeps them more liquidy. So that's what's meant by a fluidity buffer, is maintaining the fluid nature, whether the temperature is high or whether the temperature is low. So that's a major thing that cholesterol does in our membranes. All of this slide is talking about membrane fluidity and what contributes to that. Okay, well, where does the membrane come from that's going into your plasma membranes? And you know this already. Let me help remind you. But if you've been learning the material in this chapter up to this point, then you know this. So take a look at the figure that I've got embedded on the bottom right-hand corner of the slide and trace the pathway that membrane materials follow from their synthesis, from where they're made, onward to where the membrane's being deposited into the plasma, or into the plasma membrane. So, green asterisks, boom, 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 boom. Membrane components, like lipids, phospholipids, cholesterol, and proteins, including glycoproteins, are synthesized, you know where, the endoplasmic reticulum. Those are our factories where these organic molecules are being made. They are shipped in transport vesicles to the Golgi complex, where those molecules are refined, modified. Those molecules are sorted out based on where they're going to go, and then they're distributed to their final destinations. You've learned this before. And that's the pathway. You can see it on this figure. So after vesicles are leaving the Golgi complex, those vesicles are traveling to the plasma membrane and fusing to the membrane, becoming part of the membrane, and that's where the membrane comes from. So the vesicles, when they collide with that plasma membrane, they basically turn inside out, and the phospholipids in those vesicles just kind of flow right into the plasma membrane. And proteins might go into the membrane. Other proteins, secretory proteins like hormones, or maybe digestive enzymes, would be released to the outside of the cell. So there's the pathway. You should be able to describe this pathway. It's a really important pathway to show your understanding of where materials are made in eukaryotic cells and how they're distributed and how our plasma membranes get those materials through this pathway. All right, well, let's come back to this topic of selective permeability. We've just gone over the structure of our membranes, how they're put together, and their characteristics. Nail that down. So back to selective permeability. Our fluid mosaic model, remember what that means. Membranes are a fluid of phospholipids with a mosaic pattern of proteins scattered around. Our fluid mosaic model helps explain how membranes regulate or control the molecules, the substances that are going in and out of cells, molecular traffic. So in other words, tapping into that one of those concepts from chapter 1, emergent properties. The emergent properties of membranes, like what they do, how they work, those emergent properties result from the integration of these components, how these components are working together. That's consistent with the topic of emergent properties. Like a computer works the way it does because of the way the computer components are integrated. A car works the way it does because of the way the drivetrain and the transmission and the engine and everything is integrated to make the car work. So plasma membranes or cell membranes work because of the way the components are integrated. Okay, well now let's take a close look at what can cross our phospholipid bilayers and what cannot. Really know this. First, nonpolar hydrophobic molecules like hydrocarbons, carbon dioxide, oxygen, those are nonpolar and hydrophobic. They readily move through the phospholipids. They have no problem going from the watery environment through the phospholipids to the other side. So, phospholipid bilayers are highly permeable to nonpolar substances. Just like a doorway is highly permeable to a chair, okay? Highly permeable to nonpolar substances. When we move into the realm of polar molecules, then they start to have a little more difficult time. Okay? Water is very small, so it can slip through some of the phospholipids sometimes, but remember those aquaporins? If a cell really needs to move water in and out quickly, then it's not going to just rely on the water moving through the phospholipids, because the polarity of the water molecules makes them stay more on one side of the membrane or the other side of the membrane. Now, if we bump up the size of those polar molecules, make them bigger, like sugars, those are bigger than waters, they're going to have a more difficult time. In fact, they really can't get through. It'd be very rare that a sugar could get through. And then finally, ions cannot get through at all. Now, really listen to this. Ions are not big. They're tiny, like one atom. A chloride ion is just one atom, but it's got a negative charge, and that negative charge makes it stay out in the watery environment where it's attracted to the slightly positive hydrogens of waters, and they don't cross the phospholipid bilayer. Same with a potassium ion or a sodium ion. They're positively charged. They're very tiny, but the charge, this is what I really want you to remember. The charge is what makes them impermeable. The membrane is impermeable to those ions. So there's your scale. Nonpolar things can get through easily. Small polar things can get through somewhat. Large polar things, not very often at all. They really can't cross. And ions, forget it. So the only way some of these other things can more easily get through, which we'll look at more carefully, is with transport proteins, specialized proteins embedded in the membrane that allow specific substances to cross, like water, like aquaporins. But there's many others that are very specific to certain substances. Selective permeability, and what can get across and what can't get across phospholipid bilayers. This is what you need to understand here. So now let's get to this topic of diffusion, how substances cross membranes. That's what the rest of this chapter is mostly on now, is how substances cross cell membranes. And the first topic is simple diffusion. So diffusion, it's called simple diffusion, and the idea is pretty simple. If a substance is not equally concentrated in space, it's more highly concentrated in one area than another area. There's more of that substance here than there is here. Then these molecules, these substances, are randomly moving. They're bumping around and hitting each other from heat in the environment. But if there's more of them here than there are here, then overall more of them are going to be moving from high to low concentration than from low to high concentration. So just by sheer numbers, more will move from where they're more highly concentrated to where they're less concentrated. And that overall movement from high to low concentration is diffusion. That is diffusion. Imagine if we were in lab upstairs. I'm recording this in my office. If we were in lab upstairs, I could pull out a beaker. In fact, I could even do it in my office, but this would take too long on a video. And have a beaker full of water. You could do this at home. If you have some colored dye, like you use for coloring eggs or something, and just put a drop of that dye in the water. Bloop. Make sure the water's been sitting in the glass or the beaker for quite a while, so it's not like all agitated. Bloop. Drop it in the water. That dye will sink to the bottom, and just leave it there. Don't touch it. Just leave it there. Come back after about 30 minutes, take a look, and you'll see that the dye is spreading out in the water. It's spreading out from where it's more highly concentrated, that drop of dye that was put in there, to where it's less concentrated. And if you come back an hour later, you'll see it spread out even more. And eventually, molecules of any type will become evenly distributed, moving around randomly, evenly distributed, and they'll be going back and forth equally. And that's called dynamic equilibrium. And when a substance gets to dynamic equilibrium, there's no more diffusion. There's no more moving from high to low. It's all evenly spaced out, evenly distributed. That's dynamic equilibrium. So the figure in the middle of the slide is kind of showing you this. You've got those little yellow spheres on one side of a membrane and purple spheres on the other side. Each of them has a concentration gradient. And then they're able to move back and forth across the membrane, so they each diffuse in their own direction from high to low. And eventually they get to that dynamic equilibrium. Now, diffusion is a form of passive transport. And all that means is that a cell doesn't have to expend or use any energy to make it happen. It's just the energy from the molecules already vibrating from heat. That's it. That's passive. So here's another example of passive transport. Get on a bicycle on the top of a hill. Coast down. You don't have to pedal. You don't have to expend any energy. You will move passively from the top of the hill to the bottom of the hill. Passive transport. And that's what diffusion is. Cells don't have to expend any energy to make this happen. It's just the random movement of molecules. So I think I have a video I may have had you watch in doing your reading questions, but I do have a couple videos in the Canvas module that I made using a whiteboard that kind of shows this. So you could take a look at that, but hopefully you're following this. Simple diffusion. Substances moving down their concentration gradients from high to low concentration until they get to dynamic equilibrium. Aha! Well, look at that. I had a slide all ready to go where this narration is in Canvas, and I put a link in here to the video, which is in YouTube. So hopefully the link works. So watch the video, take notes if needed, but I'm trying to show you on a whiteboard, like I would if we were in a lecture room, how diffusion works. So hopefully you can check this out before you move to the next slide. If you have troubles, then, well, hopefully I can troubleshoot the problems. Okay, on to the next slide. Okay, let me get rolling here. I got to get to the right slide, catch up. Here we go. Section 4.11, moving on, facilitated diffusion. So we just talked about what diffusion is. That's when a substance moves from where it's more highly concentrated to where it's less concentrated, just by the random movement of molecules from heat. Facilitate. To facilitate means to help. So if somebody is struggling to cross the street, going across a crosswalk, and there's a bunch of traffic coming, you might run up to them and say, hey, let me facilitate your crossing of the street. Let me help you. No matter what job you might choose to do in your life, you're probably going to be facilitating or helping people in some way. If you're a nurse, then you're facilitating their health and well-being. So anyway, that's what facilitate means. So there are many substances that cannot cross the plasma membrane, the phospholipid bilayer. They can't cross through those phospholipids. But the substance, because maybe if it's charged, like an ion or a polar molecule, but it needs to get across from where it's more highly concentrated on one side to where it's less concentrated on the other side. And so the solution to that is that cells put special proteins, special transport proteins into the membrane. They are transport proteins, and they are facilitating or helping the substance to move across. There's two kinds of transport proteins that are helping with facilitated diffusion. One type is a channel protein. So take a look at the upper figure. It shows a protein with a tunnel through it, throws a corridor, a channel, and the substance is moving from where it's more highly concentrated on one side of the membrane to where it's less concentrated on the other side. And so that protein is just providing a corridor or a channel for the substance to move across because the substance cannot get past the phospholipids. The other kind of transport protein that helps with facilitated diffusion is called a carrier protein. And that's shown on the bottom part of the figure. So when carrier proteins, a substance bumps into the protein and it sticks to it. It attaches to the protein. And when it sticks to it and attaches to it, the protein changes its shape and moves the substance to the other side. Okay? So a substance sticks or attaches to a protein, and then the protein changes its shape and moves it to the other side. And so it's carrying it. That's why it's called a carrier protein. These are the two kinds of proteins, transport proteins, that are involved in facilitated diffusion, helping substances get across from high to low concentration because they cannot cross the phospholipid bilayer. Now, there are many diseases that have to do with a person or an organism not having a certain transport protein because genetically they've inherited a mutation in their DNA, and that mutation makes a protein, but the protein doesn't work. And so if it's a transport protein and it doesn't work, well, then they won't be able to transport or move that substance across the membrane. So one example is a disease called Cystinuria. And this is where a person is unable to transport the amino acid called cysteine and some other amino acids across the membranes of kidney cells. Kidneys are what are filtering your blood. And so this results in kidney stones, which can be very painful. So it's a critical transport protein, and if it's absent, if any critical transport protein is absent, then that's an inherited genetic disease. So just to wrap this up, diffusion is the movement of substances from high concentration to low concentration. Facilitated diffusion is when a transport protein, either a channel protein like an aquaporin, or a carrier protein like the one used for cysteine in your kidney cells, is helping a substance diffuse across a membrane from high to low concentration because it cannot get across the phospholipid bilayer. So this should conclude part 4 of chapter 4. And I'm dividing up these last sections into some smaller parts to finish up the chapter to help you work on them in smaller little chunks. So this ends our discussion of diffusion and membrane structure.